Welcome to my lecture on the major histocompatibility complex MHC, and the human leukocyte antigen system HLA, which together represent an antigen system that mainly plays a role in the immune response. I will focus here on the cellular functions rather than the precise divisions within this system. We will deal generally with the main histocompatibility complex and the basic classification one can make about it, going through MHCI and MHC2 and discussing the individual functions of these antigenic subgroups. I will also say something about the clinical relevance in general and draw a brief conclusion at the end. The main histocompatibility complex is an antigen system which, as the name suggests, is expressed primarily on tissue but also on leukocytes. We already know antigen systems from blood group serology that are typical of erythrocytes. In contrast, the antigens of the MHC system are expressed on all tissues except erythrocytes. The MHC is divided into the subgroups MHCI and MHCII, since these have functional differences. The various antigens in the major histocompatibility complex ensure the chemical individuality of an organism. This means that your own tissues are compatible with each other in the same organism. However, when tissues are exchanged as part of a transplantation, rejection reactions can occur between the donor tissue and the recipient if these antigens are not compatible with one another. We already know this from transfusion medicine. Of course, the actual function of the antigens in the MHC system is not to enable transplantations, but rather what is known as antigen presentation. What is behind this, we will now discuss in detail. The antigen presentation is the task of the two molecule groups MHCI and MHCII. In general, its function is to present small cell components, coming from inside or outside the cell, to the immune system. Almost every cell in the body can function as an antigen-presenting cell. In particular, mucous membrane cells, which have a lot of contact with foreign antigens, take on this task of adapting our immune system. The classification into MHCI and MHCII basically says whether the components to be presented come from inside the cell, e.g., come from viruses or tumors, or whether they are external components of bacteria or parasites, for example. Other components that come from inside the cell can also be normal protein residues and any kind of degradation products of the cell's own metabolism. Defective and excess proteins are also presented to the immune system, at least in part. In the following we will refer to all these residues of proteins or pathogens as antigens and discuss their interaction with the MHCI and MHCII molecules. The MHCII group is further subdivided in the HLA system into HLA-A, B, C, E, F, G. In order to understand the function in antigen presentation, however, we initially stayed with the term MHCII. This group of molecules is used to present the extracellular antigens, i.e., primarily the antigens from foreign organisms, such as e.g., bacteria and parasites originate, or also from particles that have entered the organism via a wound, in order to draw the immune system's attention to these foreign antigens. Such antigens are presented by means of MHCII molecules to the so-called T lymphocytes. T lymphocytes are special leukocytes that are formed in the thymus and, when they are formed, circulate in the body. As a kind of police force, the T lymphocytes are activated and then initiate further steps in the cells of our immune system, including B lymphocytes, macrophages, monocytes, dendritic cells, appendix, lymph nodes and spleen. MHCII is strongly expressed in order to process the foreign antigens that accumulate there in so-called immune-privileged tissues, such as the brain, retina, muscles, islets of Langerhans and spermatocytes. On the other hand, MHCII molecules are expressed only slightly, since a defective MHCII molecule can also cause an autoimmune reaction, and the organs mentioned are characterized by low expression of MHCII is relatively rarely affected by autoimmune reactions. Of course, the brain, retina, etc. can therefore also react less well to pathogen antigens. Let's start with antigen presentation via MHC2. There are different ways in which antigens can enter the cell, either passively through endocytosis or through active entry into the cell. After they have entered the cell, viruses, bacteria, fungi or parasites are transported to the lysosome by means of endosomes, which are small transport vesicles. The lysosome is a vesicle filled with enzymes in which the pathogens are broken down into their individual antigens. An antigen can therefore be a component of a capsule, a cell membrane, or a protein which originates from the corresponding pathogen. The antigens, which are shown here in blue, are brought into the endoplasmic reticulum by means of further transport vesicles after they have been broken down by the red enzymes. Various proteins are found in the plasmatic reticulum, including the MHC2 molecule. Here, MHC2 forms a complex with the antigens, i.e., the antigen binds to its intended location in the MHC. The endoplasmic reticulum is responsible for the production of proteins. Proteins leave the cell from the endoplasmic reticulum via the so-called Golgi apparatus. Together with the Golgi apparatus, the endoplasmic reticulum forms a kind of cell station. The MHC2 molecule is transported out of the endoplasmic reticulum to the cell membrane in a complex with the antigen. The complex of MHC2 and the antigen of the pathogen is turned outwards at the cell membrane, so that from now on the antigen sits on the outside of the cell on the MHC2 molecule. If the antigen is on the outside of the cell membrane, our body police come into play, the T lymphocytes, which are responsible for recognizing foreign antigens, bind to the complex of MHC2 and antigen. T cells have a special receptor, the so-called T cell receptor, or TCR for short, with which they bind to this complex. If the T cell comes into contact with a pathogen antigen, the T cell is activated. This T cell, which has not seen any pathogen antigen before, will from now on be able to react specifically to this pathogen. Their T cell receptor was previously variable and is specially adapted to the pathogen antigen after this first contact. If there is no foreign pathogen antigen on the MHC2 molecule, or if it is an antigen that is part of the normal metabolism of the cell, the T cell is not activated. For this purpose there are e.g. special MHC molecules on egg cells, which are not loaded with antigens. These serve the purpose of protecting the egg cells from rejection reactions by T cells. Here we see in the top left of the picture how the MHC molecule with the antigen on it binds to the T cell receptor. The T cell receptor undergoes both a mechanical change and the DNA that encodes the proteins for the T cell receptors is altered. The title receptor is therefore from now on specifically targeted to the antigen, and even if this T cell divides in the future, the daughter cell will be targeted to this specific antigen. Activation of the T cell receptor then promotes proliferation, that is, the division rate of the cell as well as the survival time of the cell itself. At the same time, the cell's metabolism will be boosted for further reactions. Here we see again in simplified form how the T cell receptor is specially adapted to the antigen after contact with the antigen. In the future, a foreign antigen no longer has to be complexed with an MHC2 molecule in order to bind to the T cell receptor. A free antigen of the same type can also bind to the T cell receptor in the future and start the immune reaction. We see our activated cell here in pink on the right side. Through contact with an antigen and the MHC2 molecule, it has a specific molecule that will be developed in future for the identification of pathogens. This so-called T helper cell also proliferates Th0 cell further into a Th2 cell. This Th2 cell further divides into two plasma cells, which respectively produce antibodies against the antigen. The antibodies formed bind to the antigens of the pathogen and mark it for attack by other immune cells. And as long as there are antigens presented to the T lymphocytes will proliferate into plasma cells, which produce antibodies. At the beginning of an infection, IgM Antibodies are first formed, which bind many antigens at the same time and are still relatively unspecific. In the course of the infection, the so-called isotype switch occurs. If there are no more antigens in the blood, the activated Th0 cells will no longer proliferate into Th2 cells but into Th1 cells. The difference is that Th1 cells remain in the body as T memory cells without developing into plasma cells. The T memory cells continue to retain their specific receptor for the antigen and can proliferate into Th2 cells and later into plasma cells if they come into contact with a corresponding antigen again. At this point it can be assumed that there are sufficient antibodies against the relevant pathogen in the blood and no further plasma cells and antibodies are needed. In view of the fact that we need MHC2 molecules for the presentation of antigens in an infection, it is not surprising that a mutation of MHC2 molecules results in a poorer immune response. In particular, infections caused by extracellular pathogens such as bacteria, fungi or parasites can be combated more poorly by the body due to a corresponding defect in MHC2 and a protracted and serious immune reaction may occur. On the other hand, a defect in the MHC2 molecule can ensure that the immune system mistakes our own body cells for pathogens and causes a so-called autoimmune reaction. In an autoimmune reaction, the body's own cells are attacked by T lymphocytes and other immune cells, which can result in various clinical pictures. We have listed some possible diseases here. In rare cases, an MHC2 mutation can also cause increased resistance to certain viruses, since MHC2 molecules of the HLA-B27 subtype have a special affinity for the antigens of HIV and influenza viruses. So far we have learned how foreign pathogens penetrate the cell from the outside and are broken down into their individual antigens. The individual antigens are complexed with MHC2 and presented to the outside of the T lymphocytes. Plasma cells then form from the T lymphocytes, which form antibodies against the specific pathogen. From the way of the MHC2 molecule we now come to the way of the MHC1 molecule. This is responsible for the presentation of intracellular proteins to the outside. These can be completely normal proteins of the cell or virus proteins, or tumor proteins that have been in the cell for a long time. In this way we will get to know other cells of our immune system and also see what the body does to fight tumors. As we already know from the MHC2 molecule, MHCI molecules are also further classified in the HLA system. MHC1 molecules also present various antigens to the outside. However, these antigens are not found in our Th0 cells, but in so-called natural killer cells and cytotoxic T cells. We find many MHC1 molecules in the organs of our immune system and, interestingly, also in the Langerhans cells of our pancreas, probably to ensure our insulin production. As with MHCI, little MHCI is formed in the brain, retina and muscle in order to reduce autoimmune reactions. In the metabolism of a healthy cell, a large number of proteins accumulate from time to time that are no longer needed. A small protein called ubiquitin is attached to excess proteins. Proteins marked with ubiquitin are broken down into smaller components by enzymes in the so-called proteasome. We also treat these components here as antigens. Foreign proteins, for example virus proteins or unusual tumor proteins, are also introduced to the antigen presentation in this way. In the next step, MHC molecules are again complexed with our species. This time, it is about MHC1 molecules, which are presented with the different antigens on the outer wall of the cell. A transport vesicle is again responsible for transporting the endoplasmic reticulum to the cell membrane. This has the name transporter associated with antigen processing. As soon as the antigen with the MHC1 molecule is on the outer cell membrane, a cytotoxic T cell can bind to this MHC1 molecule with its T cell receptor. Like The Th0 cells, the cytotoxic T cells, also have a special receptor with which they can distinguish foreign antigens from their own antigen. In the case of defective tumor proteins or pathogen antigens, these antigens activate the cytotoxic T cell. If there is an antigen on the MHC molecule which is part of the normal physiology of the cell, the function of the cytotoxic T cell is inhibited. If it is a foreign pathogen antigen, the cytotoxic T cell is activated. In addition to the cytotoxic T cells, natural killer cells can also bind to the MHC1 molecule. Natural killer cells are responsible for detecting tumor cells. Tumor cells mostly do not develop MHC1 molecules or only very few. If there is no MHC1 molecule on the cell, the natural killer cell is activated and destroys the tumor cell. Under normal conditions, it may be that no antigens are presented to the outside on the MHC1 molecule, so the MHC1 molecule is empty. In this case, an empty MHC1 molecule inhibits the activity of the natural killer cells and the presenting cell is spared. An activated natural killer cell or cytotoxic T cell produces various enzymes to destroy the presenting cell. The presenting cell, which here either has intracellular pathogens or is a tumor cell, should ultimately no longer multiply. These enzymes are various proteases or other hydrolases which can destroy proteins, DNA and other cell components. By perforating the cell membrane with perforins, water enters the cell and the target cell swells and bursts open. The remaining enzymes, such as so-called granzymes, then break down the individual components. Various defects in MHC1 molecules play a role nowadays. Beta-2 microbe globulin, for example, plays a role as part of the MHC1 molecule. If there is a mutation in beta-2 microglobulin, we find this in the blood in various leukemias, including lymphatic leukemias or in multiple myeloma. These defective MHC1 molecules are more difficult to detect by natural killer cells and the corresponding tumor cells are therefore less able to be combated. Here, too, an MHC1 defect can cause many autoimmune diseases. Defective MHC1 molecules can appear as foreign antigens to our natural killer cells or cytotoxic T cells, and the antigen-presenting cells are thus destroyed. With this we have become acquainted with both ways of antigen presentation in epithelial cells, once the way of the extracellular antigens by means of MHCII, and the way of the intracellular antigens by means of MHCI. We can now also functionally assign our body police to the various lymphocytes. In addition to the antigen presentation in epithelial cells, there is a very special type of cell in our lymphatic organs, which can combine the extracellular and the intracellular way of antigen presentation. In our lymphatic organs, these so-called dendritic cells are constantly in contact with the surrounding cells and foreign antigens from our lymph fluid. Through their long outgrowths, they can take up antigens from neighboring cells or from the lymph fluid and then present them. They have the special ability to present both extracellular and intracellular antigens to the outside by means of MHC2 molecules. These so-called professional antigen-presenting cells are therefore another central component of our immune system. What did we learn? The antigens of the major histocompatibility complex are divided into MHC1 and MHC2 based on their function. The individual subtypes of the MHC1 or MHC2 molecule are further classified in the human leukocyte antigen system. The various antigens play an important role in the tissue compatibility of our own cells. Our immune system can use it to distinguish our own cells from foreign cells. MHC1 and MHC2 molecules are important to them for the presentation of pathogen or tumor antigen to our lymphocytes. Lymphocytes then take over the task of destroying tumor cells or affected cells. Defects in MHC molecules can be responsible for many autoimmune diseases and are of diagnostic and prognostic relevance. In conclusion, I would like to recommend Marcus Ferencic's Compendium of Immunology to anyone who is further interested in immunology, as well as the immunological standard work Janeway's Immunobiology. Thank you for your attention and I hope you all learned something.